we'll get started, and next Monday is a holiday, so we actually have class on Tuesday afternoon. And then next Wednesday, we'll actually be making up one of the core visits to the genomics core. So I'll be sending out announcements about that later on. So today our first speaker is John Schiller. He got his bachelor's degree at the University of Wisconsin-Madison, his PhD from the University of Washington in Seattle. He came to NCI in 1986, and subsequently he became a senior investigator. In 1999, he became chief of the neoplastic disease section. It's in the Laboratory of Cellular Oncology. His title, Vaccines to Prevent Oncogenic HPV Infections. John.
Thanks, Terry. So on the syllabus, it says I'm going to be talking about cervical cancer, and I've taken the liberty to expand a little bit. I'm going to talk more generally about HPV-associated cancers. I'm not a cancer biologist. I'm a virologist. And so the view you're going to get is going to be kind of skewed towards virology a little bit. But certainly HPVs are a very important cause of cancer. And I think any cancer biologist should know a little bit about HPV. So in the first part of my talk, I'll talk a little bit about the association between HPV and cervical and other cancers as well a little bit, but mostly focusing on cervical cancer. But then I'll hopefully convince you it's not enough to know about a cause of cancer, the etiological agent that's overwhelmingly responsible for cancer. You have to do something about it. I'll tell you about developing the HPV prophylactic vaccines, which I'm sure you've all heard about. But then in the final part of my talk, I'll hopefully convince you it's not enough to have a good intervention. You have to actually get it to the people who need it. And I'll be talking to you about some of the implementation issues that are associated with the rollout of these vaccines. So the reason usually when we think about HPV and cervical cancer should be obvious from this slide, both in terms of the total number of cases worldwide and the etiological fraction. Cervical cancer really dominates the HPV-associated cancer scene. But it is associated with the majority of anal cancers, cervical vulvar cancers, about half, minority of penile cancers, and almost half of the oropharyngeal cancers. Overall, HPV is thought to cause about 5% of all cancers in the world. So this is really one of the major causes of cancer as an individual etiological agent. Now, there's a whole lot of HPV types, but fortunately for those of us developing vaccines, 2 types cause the majority of cancers worldwide, 16 and 18. It doesn't matter what portion of the world you look at. But then after that, the other types that are 2, 3, 4, 5, 6, 7, tend to vary a little bit, but there tends to be a certain cluster of about 5 types. Now, overall, HPV is associated with around 16 or 18 causes about 70% of cervical cancer. It also causes most of the other HPV-associated cancers, about 90%. But what this slide mainly shows is that the distribution of cancer types in the United States is quite differently skewed than it is worldwide. And there's 2 reasons, and what you'll notice is that it's not so dominated by cervical cancer. If you look at the incidence of anal, vulvar, vaginal, and oropharyngeal together, it's equal to about the level of cervical cancer, and that a relatively large minority are now in men and not in women. And the reason for this is twofold. First of all, Pap screening has reduced the incidence of cervical cancer by probably at least 80% in the United States and other countries that have well-organized Pap screening programs. This isn't the case in the developing world where 85% of cervical cancers occur. And secondly, there is what's been called an epidemic of HPV-positive oropharyngeal cancer with more than a 2-fold increase in the last couple of decades. And so for these 2 reasons, HPV non-cervical cancers are tending to be higher amounts and also more in men than previously thought. And obviously this has implications ultimately about who you want to vaccinate. Now the cancer-causing types, incidentally, just happen to cluster in one phylogenetic group over here that are separate from the types that cause genital warts, which are 6 and 11, and separate groups called hand and foot warts, And then there's a whole other bunch of groups that cause asymptomatic infections. Now, all HPVs and all papillomaviruses that infect animals have a very characteristic lifestyle, and they productively infect only stratified squamous epithelia. So this is the only tissue in which they can replicate, where they have to infect the basal epithelium, and during differentiation, they end up making their virion proteins and assembling the virus only in terminally differentiated cells. Presumably the reason they do this is to escape immune evasion. As I'll show you in a minute, their virion proteins release their capsid structures are very immunogenic. And by expressing them only in these terminally differentiating cells, they're no longer under immune surveillance. But the infection is maintained in these basal cells down at the bottom here, which are under immune surveillance, and then you only get a certain subset of genes expressed at very low levels. This is just an example, both morphologically and histologically, at an HPV-16 lesion that potentially could go on and cause a cancer. And you can see it's very inapparent here. You barely would notice this lesion. It's not like a big exophytic wart that you would see here. And histologically, this is the normal tissue over here where you only have proliferation as demonstrated by MCM, which is induced by E7 in a marker of proliferating cells down here in the basal level. But you can see you get proliferation of superbasal cells up here, and then expression of later proteins only in these terminally differentiating cells way up here. Now we can now understand cervical carcinogenesis in HPV-associated carcinogenesis by other types, for other cancers, although it's not quite as well understood, from the point of view of HPV infection. So if you look at what's called mild dysplasias, or if you, in a pap smear cytology, low-grade squamous interepithelial, or cervical interepithelial neoplasia grade 1, these mild dysplasias are just manifestations of normal HPV infection. Some infections, you see these, some you don't, and most of them just go away. But if you have persistent infection, you can get progression to intermediate moderate dysplasia, to severe dysplasia, carcinoma in situ, and eventually cancer in this process. Now, an interesting feature is that these higher grade lesions no longer produce viruses because they don't have the signals to produce the late proteins. And so this carcinogenic progression is as much a dead end for the virus as it is for the host. There's no tumor virus that wants to cause cancer. It's not part of any normal virus life cycle. It's always an aberration. Now, together with the lack of virion production, there's a stage where you tend to get higher levels of two of the viral proteins called E6 and E7, which are the major oncoproteins, and oftentimes viral integration. Now, the way these two oncogenes work is that E7 is mainly responsible for inducing aberrant proliferation in these terminally differentiating cells that normally wouldn't be making their own DNA. But this aberrant proliferation, there's a problem with this because it induces apoptosis. So it needs to have another gene called E6, which blocks this apoptotic process, leads to immortalization, and together they destabilize the genome so that both the direct activities of E6 and E7, which I'll discuss in a minute, and also this indirect induction of genomic instability leads to transformation and oncogenesis. Now the question arises, why would these viruses have these oncoproteins? I told you that the cancers don't make any virus, so why would they have these genes? Well, these viruses, like other oncoproteins as well, especially these viruses, have this issue of they're trying to get the cell to replicate their DNA in cells that are terminally differentiating, and so that they don't want to have DNA synthesis. And so by having these genes, they turn on the DNA synthesis machinery to get their own DNA genome synthesized in cells that otherwise would not do it. So just to give you sort of a summary, this is a very complicated slide, but E6 and both E7 interact with a number of different cellular partners. And the most important one, and the one that's been best studied, is the interaction with P53. It interacts with P53 in ubiquitin ligase called E6AP, which leads to ubiquitin-dependent degradation of E6. It also interacts with P300. And by interacting with the transcription complex for H-TERT, it actually can contribute to immortalization. It induces this catalytic portion of telomerase. It also interacts with anti-apoptotic factors like BAX and BAC and IRF3 and caspases, and also cell adhesion proteins through FAC kinase. E7, as I mentioned, is the main protein that induces this cell proliferation. It does so primarily by interacting with P53 or these other pocket proteins, P107 and P130. And by doing this, either by just the interaction or also induces some degradation of these proteins, it frees up E2F, which is the major transcription factor that leads to expression of cyclin E, cyclin A, and leads to S phase entry. So those are the major functions, and when this sort of thing goes awry is when we get cancers. Now, one of the interesting features about cervical cancer is where it arises. So these infections occur throughout the genital tract, both in the vaginal tract and various portions of the cervix. But almost all the cancers arise at one point, which is called the transformation zone, which is a site where you go from the stratified squamous epithelia to simple columnar epithelia in the cervix. And this is a very interesting portion because this one little portion here, it's now got markers. It's actually one of the markers is keratin 7. And these cells, right at the, you see how this is stratified squamous, now this is simple columnar? There's a very small region here that remains in an embryological-like state. And for some reason that we don't understand yet, but it's a very important cell biological question, these cells are particularly susceptible to carcinogenic progression by HPV E6 and E7. So the chances of, if you get infected in any other place in the cervix or the vaginal tract, of that going on to cancer is actually quite low. And this is one of the reasons why if in treatment you remove these transformation zone, these cells don't repopulate, they're gone. And that's why if you do ablative therapy of an HPV lesion, you drastically reduce a woman's risk of getting cervical cancer, even by a new type. So interestingly, anal carcinomas also occur at the transformation zone, whereas HPV infections of vulvar and vaginal and penis, as I mentioned, are very common, but cancers at these sites are relatively rare. And we think that they're rare is because these tissues, fortunately, don't have transformation zones. So that's why penile cancer is so much less than cervical cancer. It's not like penile infections are rarer than cervical cancers. That's not the case. This is just to show you what histologically, what this, or anatomically, what this looks like. So the transformation zone is where you go from red to this. In this area that looks whitish, it's stained with acetic acid, is where the lesion is occurring, and then this is where the cancer eventually develops. Now, the time course of HPV and its association with cancer is very characteristic in that HPV infection is very common in young people, and most of the infections are cleared spontaneously, presumably by an immunological mechanism. We think that T-cell immunity is mainly responsible for clearing lesions once they develop. And the lifetime incidence is very high. We think probably more than 80% of women get some sort of cervical HPV infection during their lifetime. But as I said, these spontaneously go away, and then this eliminates the risk for infection. It's persistent infection that's a risk for cervical cancer. Transient infection that goes away is not a risk for cervical cancer. But in a small proportion, you get persistent infection, and especially if it's 16 and 18, this is by far the single most important risk factor for progression to precancer and cancer. Cervical cancer without HPV infection is very, very rare. So moving on to the vaccine, so now we have the situation where we have a cancer that's caused by a particular subset of viruses. Obviously, the thing to do, if you want to prevent that cancer, is to prevent the initial infections that have a chance of going on and causing that. And the solution is something that we started working on back in the early 90s, 1991, 92, And what we found is that if you take a single protein from this virus, it's called L1, it's the major capsid protein. In this case, we expressed it in insect cells, that it spontaneously assembled into what we call virus-like particles, which essentially are just the outer shell of the virus. They don't have the viral guts. The only protein that's expressed in the system is this one virion protein, none of the early proteins, none of the transforming proteins. And so they form these structures that not only look like virus, but most importantly, initially we injected them into animals, and the sera that came out of those animals had sky-high activity at preventing infection by the virus of tissue culture cells. And so we thought that this would be a good vaccine because it was non-infectious, non-oncogenic, and induced very high titers of what we call neutralizing antibodies. Neutralizing antibodies prevent infection in tissue culture cells. And the key to developing these vaccines really was an observation that was made before we got involved, that if you do intramuscular injection of virions where you don't get any productive infection, you get very strong protection from experimental infection of the skin in animal models. But if you denature L1 or use L1 peptides, linear peptides, you get no protection. And so the idea is that you needed to generate immunogens that were conformationally correct with regard to this L1 to induce neutralizing antibodies. And that turned out to be the case. So based upon these findings, 2 companies took up the gauntlet to develop HPV vaccines. This was in about the mid-90s. And there are now on the market 2 different vaccines which are based on the same concept of these virus-like particles, but the specifics of the vaccines are somewhat varied. So GlaxoSmithKline has a vaccine called Cervical, which is bivalent. It contains VLPs of 16 and 18, which again are responsible for about 70% of cervical cancer. It's made in insect cells and basically the same production system that we initially pioneered. It's got a proprietary adjuvant, which they call ASO4, which contains a simple aluminum salt and something called monophosphoryllipid A, which is a detoxified form of LPS. And this is actually quite a milestone because it's the first case where a TLR agonist has been licensed for a prophylactic vaccine in the United States. Previous to this, all vaccines just had either nothing or simple aluminum salts. So this is quite a breakthrough in terms of the regulatory industry that we're now able to use TLR agonists in vaccines. Gardasil is quadrivalent, and it contains the same 2 high-risk types, 16 and 18, but also 6 and 11, which cause about 90% of genital warts. And it's got a simple aluminum salt adjuvant, which is a little bit different than the one that's used here. And it's made in yeast, presumably because Merck had made their hepatitis B vaccine in this yeast strains. Before that, they knew how to make a lot of protein in yeast. And both vaccines are injected 3 doses over 6 months by intramuscular injection. So this slide just shows the timeline of the development of the vaccine and the parallel development of our understanding that the virus was responsible for cervical cancer. The virus was first discovered by Harold Zerhausen, who later got the Nobel Prize a few years ago for this discovery back in 1992. And about a decade later, we showed that the VLPs could be produced and induce neutralizing antibodies. It took another 7 years to have the first clinical trials, which incidentally corresponded to the first publication that said that HPV is a necessary cause of cervical cancer. It's the first time a single etiological agent has been deemed to be actually essential for getting a particular cancer. And then it was actually almost 15 years from the time we discovered the VLPs before the vaccine was licensure, first in females and then 3 years later in males. It was absolutely critical to establish the pre-malignant disease that were associated with cervical cancer, that were HPV associated, because we could never have run a trial in which cancer was the primary endpoint of the trial. One, it would have taken us decades and decades, because it takes decades for primary infection to go on to cause cervical cancer. And secondly, in active follow-up studies, you identify pre-malignant lesions, and ethically you have to remove them. You can't let anybody go on and get a cancer. And so we could never have demonstrated protection directly from cervical cancer. And so what the regulators allowed us to do is to use prevention of moderate and severe dysplasia, SIN2-3, as a primary endpoint to get cancer as an indication on the label. Because we understood so well, in part from pap smear testing, if you remove these lesions, women don't get cancer. And we also biologically understood it and could reproducibly identify these lesions. And so because we understood the natural history of disease well enough, we were able to go ahead with the clinical trials, not use cancer as an endpoint. And when the results of these trials finally came through, we were all remarkably surprised and gratified at how well these works. Because you have to understand that in the context of doing these trials, a lot of people had tried to develop vaccines against sexually transmitted diseases. And at that point, and even today, the success was zero, essentially. And so I have to tell you, when we went around to the companies to say, you know, why don't you develop this vaccine? We got all this great, you know, animal data, all this great in vitro data. They said, oh yeah, that looks really good, but sexually transmitted diseases just don't work. And so it was really, you know, it made a difference now that we actually know that sexually transmitted diseases can work. They can't only work, but they can work really great. So this is just a summary of some of the data for both Cervarix and Gardasil. So if you look at the immediate cancer endpoint, SIN3, that's a high-grade dysplasia carcinoma in situ, the immediate precursor of cervical cancer, protection was 100% in women who didn't have exposure to those viruses before they got vaccinated. That's a very important point, okay? I'll talk about women who have been exposed in a minute, but these are women who are naive to the vaccine types, okay? But again, this is a prophylactic vaccine. It's meant to vaccinate girls before they get exposed to the virus. And genital warts wasn't too bad either. It was 96.4% prevention. So this is way better than any of us had ever hoped. And I have another lecture where I just basically go over and try to explain why the vaccine works so well. And this is something that our lab has been trying to figure out, is why this vaccine works so well and all other, you know, sexually transmitted vaccines are terrible. But that's another lecture, and I won't go into that too much this time. So in terms of pre-neoplastic disease and infection, just as a summary, so we've shown that the HPV vaccines are very good at preventing infection and intraepithelial neoplasia for cervix and the anus, and also vulvar-vaginal. For the penis, it's been shown that it prevents infection, but not disease. And the reason for this is that penile disease is actually relatively uncommon. Most of the infections don't lead to visible lesions. And so this hasn't actually been formally demonstrated. There's been trends, but there's not been enough cases to formally demonstrate it. For oropharyngeal, and remember, oropharyngeal is this thing where the incidence is going up. We're really interested in this. But unfortunately, there's problems because we actually have some data that it prevents oral infections, but we don't understand the natural history of the disease well enough to know that the infections we're measuring are on the same causal pathway as the infections that cause the oropharyngeal cancers, which occur deep in the tonsillar crypts for the most part, okay? And because the premalignant lesions are actually unknown or very seldom identified, we really haven't been able to determine whether or not the vaccine prevents the intraepithelial neoplasia that are the precursors of oropharyngeal cancer. And unless a lot happens, we'll probably never really know whether the vaccine prevents oropharyngeal cancer until 20 years from now, we see a drop in the people who have gotten vaccinated. So one of the issues is two types cause 70% of cancer, but then there's a whole bunch of other types that cause the rest. And the question is then, is this vaccine type specific, or does it protect against these other types? And what's shown here is some data against 6-month persistent by one of the vaccines. You can see it's very good at preventing 6-month persistence by 16 and 18, pretty good against 3 other types, which are actually very closely related to either 16 or 18, but no significant protection against these other high-risk types. And overall, if you take all the high-risk types that cause infection in this study, and you take out 16 and 18, it only protected against 12% of those. So what we say is there's a little bit of cross-protection, but protection is type-restricted. You can't really count on getting a lot of protection on the types that aren't targeted specifically by the vaccine. And so again, this is a bit of an issue because now 16 and 18 cumulatively cause about 70% of cancers worldwide. But after that, there's no type that causes more than about 4%, okay? So how do you deal with those types? And this just shows in the United States that 16 and 18 causes about 70% of cervical cancer, but as you move into lower grade diseases, the proportion goes down. So it's only about 50% of high-grade dysplasia and only about 25% of low-grade dysplasias. So a lot of women will still be getting pap smear abnormalities that won't be necessarily likely to go on to cancer, but they're not caused by 16 and 18. This just shows actually the protection that was seen in the clinical trials, which is somewhat higher in this. And the reason for this is, one, there's some cross protection, and two, 16 and 18 lesions tend to progress more rapidly over time than by other, quote, oncogenic types. And so in a short trial of 4 to 6 years, you're going to see better protection against 16 and 18 than the other types. Now, one exciting thing that's been happening is that Merck has been developing a vaccine that includes not only 16 and 18 and 6 and 11, but the next most common high-risk types. And I can tell you that today, for the first time, the results of that study, because they just announced it today. I've actually known it for a little while, but this is the first day that I've actually been able to talk about it. So they made an announcement about a week ago that pivotal efficacy trial is complete and primary endpoint has been met. But just today, they actually announced that, so what the trial was, it was comparing Gardasil to this, what's called the nonavalent vaccine. And so for 16 and 18, there's about 100% protection probably for both of them. So it just looked at immunogenicity equivalents. But of the types that weren't in the vaccine, in comparison to Gardasil, they found 98% efficacy against these 5 additional types. So they haven't gone to the FDA yet, but looking at this data, it's quite likely that the FDA is going to say, yeah, go ahead and start giving this to girls, okay? So this is really going to help get protection against these other types that aren't in the original vaccine. Of course, one of the questions is, all of you who have already gotten the quadrivalent vaccine, what are you going to do? You're going to take the new vaccine, or are you just going to be satisfied with what you got? So, you know, it's going to be kind of an interesting issue as to see what happens with that. I don't think they're going to sell the vaccine that just has the 5 new types without the other types in it. They have run studies, safety studies, where it's safe in women who've gotten the previous vaccine. So if you want to spend your money to get the new vaccine, your insurance might not cover it, but you might be able to get it. Oops. So just in terms of what we think is happening here, we think that the vaccine is protecting from initial infection, because most vaccinees never test positive, even by very sensitive PCR technology. We don't think they're getting lesions and it's making them go away. We think that it's not infecting at all. And when we do see breakthrough, quote, in quotes, infections, they tend to appear early during the trials. Okay, so protection's better in year 4, 5, 6 than it is in year 1. Well, why is that? I mean, the antibody titers are, you know, the immune response isn't higher farther down than earlier on. So what we think were really, these early breakthrough infections are just emergence of prevalent infection. In other words, they have the infection, we haven't been able to detect it at the time of vaccination, and then it kind of comes out. They already had it before they were vaccinated. So we really think that most women actually have sterilizing immunity, which is pretty amazing. And we think antibodies are the main mediators of protection for several reasons. There's high levels of virus-neutralizing antibodies are routinely generated, and cross-protection in clinical trials mirrors the antibody-mediated cross-protection in vitro. Maybe most importantly, in animal studies, we can see complete protection by passive transfer of serum from a vaccinated to a naive animal, okay? And the neutralizing antibodies recognize these variable type-specific loops that are on the surface of the virus. We don't think cell-mediated effector mechanisms function in part because they would only work after infection. We think we get sterilizing immunity. And again, perhaps more importantly, L1 is not detectably expressed in the basal cells. So if you look again at the basal, at a virus that's virus-producing lesion, the L1 is only made in these terminally differentiated cells, whereas the infection is being maintained down here where there's no L1. So it's hard to imagine if you got T-cell responses to these cells up here, it's going to eliminate these cells. If you look at during progression, because these cells become dedifferentiated, then you get less and less and progressively to the point of no L1 expression in a high-grade premalignant lesion. And the VLP vaccines also don't increase the rate of clearance once you have an infection. This is just some data from the Costa Rican trial that was sponsored by the NCI, where if you look 6 months or 12 months after vaccination of women who already have an infection, and you ask what percentage of them clear, it doesn't matter whether you look at the vaccinees or the controls, the rate of clearance is the same. So in 12 months, about half of them have cleared, okay? So we think that this vaccine is working entirely by an antibody-mediated mechanism. T cells probably help to generate the antibodies, get switching to IgG, but we don't think that they're the effector mechanisms that are involved. And again, the vaccine has great efficacy, probably in part because it's got great immunogenicity. So this is the percent seroconversion, which is basically 100% to a rounding error. And the responses are very durable. So this is a result from Gardasil, from Cervarix. You can see that after 6 years, everybody is still seropositive. You've had, after the first year and a half or 2 years, there's a plateau. And basically, there's no change in antibody titers going out. And this has been true now up to 9 years. From about 1.5 years to 9 years, we have data that you've absolutely stabilized antibody titers. And this is true whether you look at just ELISA titers, where you look at antibodies that bind the VLPs, or functional antibodies that prevent the virus infection in vitro. And these levels are higher, at least a log higher than the levels that are generated by natural infection. So although we don't know how long the vaccine is going to last, it was, 2006 was the first data. We think that there's so far been strong protection for 8 years with no increase in the number of breakthrough infections over time. As I mentioned, most of the breakthrough infections occurred early, not late. And so given the fact that the antibody titers aren't going down, we actually are very optimistic for very long-term protection for these vaccines. And some new data, which again, the data actually was just announced today or yesterday. This is pretty good data to be here for this lecture. We're getting a lot of good new data. This is something, a paper from the NCI that we're involved with. And so in this Costa Rican study, there was some women, a subset, who got only 1 dose or 2 doses. And we continued to follow them and looked at their antibody responses. And we looked to see if they were going to get 16 and 18 infection. And the remarkable thing is that if the women who only got 1 dose had ended up with stabilizing antibody titers, and they were only 5-fold less than the titer after 3 doses out at 4 years. And protection was 100% in those women. Now, this is a really encouraging finding, but I have to qualify this, that this was not a controlled trial. In other words, the women were not randomized to get 1, 2, or 3 doses. We tried to look if there's anything difference between women who had 1 dose and multiple doses, and there really wasn't any major differences in terms of risk factors. But we're hoping to move forward with actually randomized trial between 1, 2, and 3 doses to see if we can get complete protection with 1 dose. Now, this is very surprising, and people have not normally seen stabilizing antibody titers after 1 dose of a subunit vaccine. This is something that's seen after live virus vaccines, but not subunit vaccines. But we really think it's the display of these repetitive epitopes that form this virion surface that send a very strong signal to the B cells to survive and keep pumping out antibodies the rest of your life. And so I actually am optimistic that eventually this is going to be a 1-dose vaccine. But we can't recommend anybody just get 1 dose. So don't tell any of your friends, go just get 1 dose. If I had kids right now, though, they'd get 2 doses. And a lot of the world is moving, or many countries in the world are moving to 2 doses now. Because 2 doses also gives no difference. As a matter of fact, if you give 2 doses to young girls, they get as high antibody responses as 3 doses of young adult women where the efficacy was so high. So because of this, for the younger kids who actually respond better to subunit vaccines, a lot of countries, I don't say most, but many countries now look like they're going to move to a 2-dose trial. Yeah, that's a good question. So we've shown this with Cervarix and Gardasil. It's unclear whether it's going to be as good because we don't know if the TLR ligand in the adjuvant plays a role. Okay, that's a really good question. And so one of the things we want to do is run a controlled trial where we would compare the two vaccines, probably the new nonavalent vaccine, and see whether it's a very important thing to determine scientifically for new vaccines is whether virus-like display to get persistent antibody levels you need to have a TLR agonist or whether you can get away without it. Because the future of subunit vaccines is virus-like display. I'm absolutely convinced of this. Okay? If we have tetanus or, pertussis or anything else, if you could do virus-like display, you probably wouldn't need to revaccinate. The question is, would you need a TLR agonist? Great question. So one of the questions that comes up is how do the antibodies that we think we're almost certain are protecting from infection get to the cervix? Those of you who know low immunology, you know that if you do systemic immunization, it's really good at making systemic IgG, but really lousy at making secretory IgA. And this isn't a virus that's infecting a mucosal site. And so most people think when you think about mucosal sites, you think of secretory IgA. In the gut, almost all your antibodies are IgA that are secreted there. But we really caught a break because at the cervix, about half of the antibodies are transudated IgG. They're pumped out by the neonatal Fc receptor. So we can get by with antibodies that are systemic antibodies because they're elaborated on the cervix quite readily by this mechanism. And perhaps more importantly, we've done studies where we found that in order for the virus to infect, it initially has to bind the basement membrane that separates the dermis from the epidermis and undergo a series of conformational changes before it can infect. You can imagine that if you have a microabrasion that leads to exposure to the basement membrane, you're going to have capillary and interstitial antibodies flowing out of here. And so the virus is going to be trying to infect against a gradient of antibodies, okay? And we think that this is the major mechanism that we're actually getting protection because we get such good protection against genital warts. And many genital warts in both men and women are on sites that are not bathed by mucus, okay? And yet this systemic vaccination is inducing very strong protection. So we think that this actually may be the main mechanism of protection. So it's got a little mess. So the conclusion of this portion, we think the VLP vaccines are highly effective at protection against a spectrum of anogenital endpoints, from incident infection to high-grade precancer. Gardasil is also highly protective against genital warts in both men and women. But protection is type-restricted, consistent with protection being antibody-mediated. And although the duration of protection is still unknown, as it would be for any vaccine that's just been recently introduced, the strong protection at 8 years after antibody levels have stabilized makes us very encouraged for long-lived protection. So now I'm going to turn a little bit more to the implementation aspects. So I think, in a relatively short number of years, but I think we can now consider these established products. They've been licensed worldwide for prevention of cervical cancer, in some cases other HPV-induced neoplasia. There's been millions of doses sold around the world. The safety issues, although people continue to raise them, so far there's not been any signal that even with this number of doses administered, that they have any serious adverse events that are associated with them. And just to point out, Gardasil was approved for genital warts in men in 2009 and for anal cancer in 2010 for both sexes, although the trials, interestingly, were only done in men. And this just is an example of globally where the vaccines are licensed, where either the quadrivalent, bivalent, or both are licensed. Incidentally, interestingly, it hasn't been licensed in China yet. China's trying to develop an in-house their own vaccine, but so far they haven't accepted approval of either Gardasil or Cervarix based upon data that's been generated in other countries. So the question is who to vaccinate, or these general implementation question. First of all, who to vaccinate? Well, clearly, girls, before they become sexually active, are the main target for this vaccine, at least worldwide, because women bear the majority of the burden of HPV infection, which is cervical cancer. But then young women might benefit if they haven't been exposed to 16 or 18. Just because you're sexually active doesn't mean you've absolutely mean that you've been exposed to 16 and 18, but it becomes the law of diminishing returns as you get older and have more sexual partners. But also boys certainly do get HPV-associated cancers, and they're the vectors for infecting the women. So one can imagine herd immunity developing faster if you vaccinate boys, even if your main goal is to prevent cervical cancer. Older men, it's probably less advantageous than the boys. Yeah, and this slide just shows why we have to vaccinate younger girls. Because this is data from the US and Great Britain that shows that within 4 months of becoming sexually active, about 20% have had an HPV infection. By 2 years, it's almost 50%. So if you wait until your daughters are already sexually active, there's a reasonable chance that you might miss the opportunity to protect them. As I mentioned, once they have the infection, the vaccine's not going to do anything for them at all. So it's interesting that the CDC, who determines the vaccine policy in the United States, initially recommended, the recommendation for boys was permissive. But in October 2011, they actually went on and recommended that you have routine vaccination of 11 and 12 and catch-up vaccination of 26. So the change in recommendation was really based upon two things. One, based on the poor uptake in girls. If you do modeling of herd immunity and you get very strong protection in one sex, you almost don't need to vaccinate the other sex. And I'll show you some actual data from that from a country in a minute. But if you get relatively poor uptake of an intervention that prevents infection, then you get more herd immunity benefits out of vaccinating the other sex as well. And the other thing that prompted them to do this is that when Gardasil was labeled for prevention of anal cancer, FDA approved it for that. Now it's really hard to prevent a benign intervention for something that could prevent somebody from getting cancer. And they just couldn't do that, even though the cost-benefit ratios are not as high as preventing infection in women. Another issue is the delivery of 3 intramuscular doses to early adolescents. And this is actually not rocket science. If you want to get high coverage, you vaccinate in schools. So this is an example where both in Scotland and England, they vaccinated 12 and 13-year-olds in schools, and they got very high coverage. And for some reason, I don't really understand the politics, Scotland continued to vaccinate or do a catch-up vaccination in schools for 17, 18-year-olds. They got high coverage. England decided they would do it in clinics, and they got 30%. Australia is above 80% in a school-based vaccination, and we're at about 30% 3-dose coverage for 13 to 17-year-olds, which I would kindly call our system disorganized. You may have worse terms for it than that, but, I work for the government. I can't say, too many, but, Anyway, let's call it disorganized. Now, it's actually possible, I didn't think this was going to be the case, but you actually can get good coverage with clinic-based, and this has been demonstrated in Denmark, but they did a remarkable job of, before they introduced it, to educate the parents, but more importantly, educated the pediatricians as the value of the vaccine and got them on board. And we haven't really done a good job of either. But this just shows what can happen if you do get good coverage of a vaccine. And already in Australia, you can see that if you look at SIN2+, which is this intermediate and high-grade cervical dysplasia, there's been a remarkable drop in incidence in under 18-year-olds, and some evidence of a drop in 18 to 20-year-olds, but no drop in these older women who weren't part of the vaccination program. Now, genital warts is an earlier manifestation of infection than SIN2-3. On average, it takes a very short period of time for an initial infection to manifest itself as genital warts. Oops. And what you can see is that in the vaccination cohorts, especially the under 21s, there's been a remarkable 92% reduction in genital warts based upon this program. And even in the somewhat older women, it's reduced by 73%. But even more remarkable, if you look at the rate of genital warts in heterosexual men since the start of vaccination, you don't need to be a statistician to see that this is pretty much dropping off the table. And the boys were not part of the vaccination program. They just started vaccinating boys. kind of wonder, well, why are they doing it? Because they're getting this kind of, they're getting an 82% reduction. And they keep hitting the wrong button. But there's no significant vaccination in the older guys, okay, reduction. And so this is really due to herd immunity. There's also no reduction in MSMs, okay? So it really is that the women are being vaccinated, protected, and preventing the infection in men. So this, I think, bodes very well for even if you don't get complete vaccination coverage, you can really control the disease if you can get the majority of people vaccinated. As long as the vaccinated and unvaccinated are in sexual cohorts and not really in separate groups. This is some data, some more from the United States, where we're actually not doing very good with HPV in comparison to two other vaccines, the tetanus, acellular pertussis, and the meningococcal vaccine that's delivered also to adolescents. This is data from 2010. You can see that it's 49% at least one dose and 32% having all three doses. And male vaccination is about 1.4%. So this was just about the time that we got vaccination recommendation, a strong recommendation from the CDC for boys, and insurance companies are now covering it. So maybe we'll start to see somewhat of an increase in vaccination of boys. And this is an update that just appeared a couple days ago. So in adolescent girls, 13 to 17 United States, 33% have had all doses. And it's basically was going up. In the last few years, it's just gone flat. And 54% have at least one dose. But, if we look at the one-dose data that I showed you on that other slide where we get 100% protection and no change in antibody titers, I think we're starting to look at the one-dose data a little bit differently than we did before. We may not consider this as much of a failure as we did before. And maybe consistent with this, if you look at the reduction in the vaccine-targeted types in 14 to 19-year-olds over the period where we've done vaccination, it's dropped by 50%. So that's very significant. It's actually closer to this number than this number. So part of that may be due to herd immunity, but part of it may be the fact that we actually, even these people are successes. But again, don't recommend people just get one dose, okay? That's not the take-home lesson here. Maybe eventually will be, but not less. But there really is a lot of missed opportunity. They actually calculated that if all the girls who came into a clinic in which they would have an opportunity to have a physician or a nurse give them this vaccine, what would the coverage rates be? It would be 93%. So a lot of the problems is that physicians are not strongly recommending the vaccines. In some cases, parents are opting out of it. So one of the issues that we have to contend with is the effect of the vaccine on cervical cancer screening recommendation and compliance. And we clearly can't give up screening because the vaccine won't help women with established infections. I've showed you that data. And since the vaccine is type restricted, it won't protect against 20 to 30% of cervical cancers. 20% is being generous that there's some cross protection. And so we really need to convince vaccinated women that they need to comply with screening programs. And it'd be too bad if we can't change screening programs eventually in vaccinated women, especially the women when they start getting the nonavalent vaccine where we get coverage for more different types. And part of the reason for this is the inconvenience, and part of it is because we just spend a ton of money on screening. So if you look at the annual direct medical cost for all the burden of preventing and treating HPV-associated disease, over 82% is spent on screening. So we really would hope that in the long run, we would be able to do something about screening in the vaccinees. And the overall idea, although the specifics are not at all clear, we're going to have to do a lot more research and evaluation to know how and when we can eventually change screening recommendations. But the idea is to go from the pap smear, which is relatively insensitive. In other words, you can be pap smear negative and still have a reasonable chance of developing cervical cancer in the next 10 years because a lot of lesions are missed. HPV DNA testing is much more sensitive. So if you are HPV DNA negative, the chances of getting cervical cancer in 10 years are really, really low. And so what we would do is switch to an HPV-based cervical prevention strategy where we would vaccinate with HPV early on to reduce this peak early on, and then the infections that do result, we would use this highly sensitive test and then probably triage to screening for those that are HPV DNA positive after age 30, but do this relatively infrequently. And in modeling, doing this type of thing could not only prevent more cancers, but do it at a lower cost than the current Pap screening technology, which is really dependent upon repeated screening because of this relatively low sensitivity of the assay. That's not to say that it doesn't work. It works. We've reduced cervical cancer by 80%. It just is expensive, and you have to have a lot of different screens in order to have it work properly. And finally, how to deliver the vaccine to the economically disadvantaged individuals where 85% of cervical cancers occur in developing countries. And really, this has got to be a multi-pronged approach. It isn't just one size fits all or one thing to do. It's encouraging that recently both companies have committed to sales to Gavi, which buys vaccines for the 72 poorest nations in the world at less than $5 a dose. This is still pretty expensive for a Gavi vaccine. A lot of vaccines cost a nickel. Another thing that we're working with is companies in emerging countries to have the vaccine manufactured there, for instance, in India and China, Brazil, other countries like that could manufacture the vaccine at a lower price. This has really been responsible for the hepatitis B vaccine going from almost $100 a dose to now most UNICEF buys the vaccine for 20 cents a dose. So, but it took 20 years to get it to there, and we're hoping that we can get the timeline shrunken for the HPV vaccine. Obviously, this has been a recurring thing, is deliver fewer than 3 doses. It doesn't take a statistician to figure if you deliver 1 dose or 2 doses at worst, it's going to cost less than delivering 3 doses. It's the type of health economics even I can understand. And the last thing I won't go into, but we're working on second generation vaccines that could be manufactured or delivered at lower cost. Well, I'll go into one of the really cool ideas that I like that we're starting to work on is to make a recombinant measles vaccine. About 70% of kids around the world get measles. In an animal models, if we put L1 in the measles vaccines, it doesn't compromise measles immunity, and we get as high a dose of antibody response from 1 dose of the measles vaccines as 3 doses of the VLPs. So you can imagine this where you get HPV for nothing. So that would be a vaccine that would be cheaper to manufacture and deliver because measles is such a standard vaccine. The problem is you can't compromise measles, or then you're substituting one bad thing for another. So I think I'm going to stop there, and in case there's a few questions, and just acknowledge the people in my lab. Obviously, I'm listing people that I've worked with directly, particularly Doug Lowy, who's been the co-director of all the research in our laboratory, some very good academic outside researchers. But also we have to give a shout out to the people in the companies who really spend an awful lot of time and effort and money and resources to bring these vaccines to market. It doesn't just take the academics on the front end of stuff, but it takes good people in the corporate world to bring a good intervention to people and hopefully prevent cervical cancer. So at that, I'll stop and take any questions. Yes. There's been some studies in HIV positive children and also adults. Now, I have to say that these are people that are on HAART, so they're not, they have reasonable T cell counts, and responses are significantly lower, but still strong, okay? So one of the possibilities would be, for instance, they may get an extra dose or something like that. But looking at the levels that they get, my prediction would be that they would be protected. And certainly, I think it's already recommended that HIV positive children, because people with HIV could have significant and much greater problems with HPV infection than the non-HIV people. that there's special emphasis to make sure that these kids get vaccinated, okay? So my guess is that it'll probably work, and then maybe they'll have to be revaccinated where other people wouldn't. But the overall response is pretty good, in part because these virus-like particles, actually, they're an example of what's called a T-independent vaccine. Because they have multivalent components, they actually can generate reasonable responses, like, for instance, in mice that have no T helper cells whatsoever. Well, yeah, I'm sorry. So for the people back home, the question was, wasn't the non-valent vaccine development challenging because of immunogenicity? And I think overall, there was not a lot of competition. First of all, none of this is published, okay? So I can't say a whole lot about this. But from a regulatory point of view, they had to show non-inferiority of immunogenicity for the types that were in the other vaccine, okay? They were very immunogenic. So let's just say it goes down a little bit because you have these other types in that. It still may be way over what we need for protection, but because it's gone down a little bit, the regulators are going to kind of go, I don't know. And so that part was challenging. It wasn't challenging to get good responses to all the types, but to actually show non-inferiority, that it wasn't even, a little bit lower, okay, that was the challenge. So that there's beyond any shadow of a doubt that the immunogenicity is the same, okay? But there doesn't seem to be like a whole lot of competition between the types for generating antibodies. That the antibody secretion is different? You mean to natural infection? We don't have any, the question is whether the protection, if I get it right, whether the protection in oral will be any different because of the transudation of antibodies. Again, we think that protection is mainly due to exudation at sites of trauma. But the oral case is really interesting because we think that the virus has to bind the basement membrane. And anatomically, the tonsillar crypt is one of the few places where at times the basement membrane is exposed, where there's no epithelium over it. So one could imagine it could go down directly. But if that basement membrane is exposed, it should also be exposed to antibodies oozing out. I mean, these are very immunological organs. And so my guess is that protection would be strong there as well. So far, the data we've looked at, it's very kind of preliminary data. And again, we don't know whether those infections are oral pharyngeal, where they are in the oral cavity. It does look like they have similar levels of protection. So just in terms of, we don't have any data, but if I would have to predict, based upon what I know about the biology, I would predict that it's likely that we would get protection. But again, it's not based upon any data. And the vaccines are not licensed for protection against oral pharyngeal cancer. We can't use it in any of the cost effectiveness modeling or anything, because it's not licensed for that indication. Do I think it's actually going to be a benefit of the vaccine? Yeah. Because one important point is that it wasn't clear before, because it's possible these oral pharyngeal infections were, for instance, acquired during birth from a mother. They just sat there for a long time, indolent, and eventually got infection. They weren't sexually transmitted, okay? But we actually think they're sexually transmitted, now looking at the epidemiology for the most part, and so that even if they don't protect from the infection process itself, if the vaccine overall is preventing genital infection, the exposure to the epithelium in the oral cavity is going to be diminished, and so indirectly it's going to protect against those infections. It just may take a little bit longer to see the protection. So I think it's going to work one way or another, directly or indirectly. Okay. Yeah. Turn this off so we're not.
Okay, our next speaker is Mukesh Verma. He got his PhD in India. Subsequently, he was at the biochemistry department at George Washington University. He came to NCI in 2006. He's in the epidemiology and genomics research, and he's in the branch chief for methods and technology branch. And the title of his talk, Cancer Epigenetics.
I was in Georgetown also. Yeah. After GW.
Or if you want to use that.
Okay. For forward. Yeah, that's fine because I can see better.
Yeah. This is the laser pointer.
Okay. Thanks, Terry, for this opportunity to discuss about cancer epigenetics. And this is a relatively new area compared to genetics. So time to time when I discuss, I will compare genetics with epigenetics. I am in Cancer Institute, and cancer is a big problem, as you see. Mostly in men, prostate cancer, and women, breast cancer, they are the number ones. And cardiovascular disease and cancer, these are the two major killers in US as well as around the world. And in 2010, there were estimated 13.6 million cancer survivors, but we expect that by 2022, there will be 18 million. So although some success we have achieved, but still it is a big problem. In major cancers like breast, cervical, colon, prostate, lung, it takes many years to develop this cancer. So our focus is that if we can identify such markers which are early, then it is better. Although in our program is epidemiology and genomics research program, we follow up after treatment also. We do epidemiology or etiological research to see which one are giving response and which treatment is good. All those trials also we do. So we keep track of survivor also to improve the quality of life. But main focus still is on how to detect early, what are the markers, whether we can use in combination or alone. Genetic was developed first, so genetic markers have been identified more, and if you see here, a different age group, different kind of markers will be there, as well as the development of cancer will tell that which markers are coming. So in some cases, in some cancers, those mutants or genes have been identified, but in some other cancers, we do not know. So in genomics, I will tell that to find out what are the variants associated with cancer, genome-wide association studies were started. And initially, 1 or 2 sites were there where they could identify in which chromosome those genes or variants are there. And that grew very rapidly because of the technology development. And by now, we have so many chromosomes and in which we have identified genomic sites. So 140 loci marked by SNPs or small nuclear polymorphism. And these are the locus that has been shown here. Now, if we see about these cancer, how the distribution of different mutations in different cancer is there. So in children, the mutations are different than in adults. So these are the frequencies given there. And after studying several cancers, Bert Vogelstein, he has come up with a model that basically 12 major pathways are there, and driver mutations are about 140. And 3 cycles are there, are 3 steps like cell survival, cell fate, and genomic maintenance. Those are the key common to most of the cancers. In epigenetics, time to time in different magazine, you will see what epigenetics can do, what is it. So it is from one German magazine where they say, the new science of epigenetics reveals there are reserve of natural happiness within your DNA. But I will tell you what is scientifically actually it is. In cancer, Initially, it was thought that it is genetic disease, but now evidence is there, it is genetic and epigenetic disease. And it is environment which makes you more prone to cancer. And as soon as environment comes, the environment interacts with epigenetic component first. To distinguish genetics with epigenetics, monozygotic twins studies were done, and then discordance was studied, how much is there in monozygotic twins, whether it is in cardiovascular disease or others, so 100% concordance was not there. So that gave the idea that there is something else also. And based on that, pharmacogenomics came for genomic side, how much do you respond for drugs, so same way epigenomics is coming, and microenvironment or microbiome, they affect genetics and epigenetics both. So genome is affected by age, diet, disease, lifestyle, environment. Epigenetics is also affected by these. But in genome, once you sequence the genome, almost it is like there, but epigenome is dynamic. It changes with the time. So if you are you want to find out epigenome sequence or other thing, you have to do many times. At the same time, in some diseases, like neurological disorders, genetic component is dominating. While in cancer and other diseases, it is more epigenetics. So those kinds of studies also, it has been done. And in Europe and US, some twin cohorts are maintained in which these studies are still being carried out. And diet and environment, they play a major role in epigenetic regulation. Another point I would like to make, that whenever some extra radiation or exposure or toxic substances are there, then few things like transcription factors or cell cycle regulatory changes, they are the first one which will happen. And then chromatin changes or methylation or histone changes, but if it is for a long time, then only genetics come. So it is epigenetics first which comes, so it is better to focus on this also. And in few cancers, those changes have been measured also, that how many translocations or deletions or amplifications, those are there in different cancers. So that has been done in genetics. Now throughout life, different changes are there. Lot of genetics has been done, and for epigenetics point of view, so in germline some changes are there, epigenetic, which are important, and then with the time they change. And initially, environmental exposure, these are important, and maternal factors, they contribute to the disease. But in adult life, it is more diet and lifestyle. And to study epigenetics, at different time, you can collect different samples and follow up. And those technologies I will be discussing. So if you see, if a smoking mother is there, then not only her life is affected, but 3 generations are affected. She herself as well as fetus and reproductive cells in 3rd generation. And when in stem cells, changes in mutation or SNPs and epigenetic changes were observed, so epigenetic changes come first. And at any stage, the number of epigenetic events are much more than genetic events. In environment, these are the different agents which affect epigenome and genome also, like arsenic. It causes genetic epigenetic, benzene, methylation profile, especially in leukemia, cadmium, hypermethylation in lung, chromium, lung cancer, nickel, and all these. Asbestos is studied more because its exposure is more all over the world. And in this, initially, mechanism was not known. It is not mutagenic. So in this, epigenetics came very much to understand that. And then some genes were identified which were hypermethylated in different cancers, especially in this lung cancer. And asbestos amount or that was carefully counted, which are called how much is the body burden. And based on methylation profiling and asbestos count, it was concluded that in asbestos exposure and others, epigenetics plays an important role. In chromosome, as you know, DNA and proteins are there, histone proteins. And nucleosomes, it is about 2 turns of DNA, 146 base pairs. And histone and DNA, they neutralize each other's charge. Basically, histones are of 4 kinds: H2A, H2B, H3, and H4. And so much about epigenetics, that these are stable alteration, but there is no change in nucleotide sequence. That's the major difference in genetics and epigenetics. You know about genetic code, but now these methylation code and histone code, those have been accepted. So what genetics provide us, that tells us what can be done. These are the blueprints of whatever cells or organism is capable of. But when it should be done, how long it should be done, when it should be stopped, that is regulated epigenetically. So as I mentioned, it's the components of epigenome. Histones are there and DNA are there. Along with that, we have microRNAs. They are part of that epigenetic regulation. So in the chromosome, these nucleosomes, the way they are positioned, if they are tight or relaxed, the expression of gene is affected. Histone and non-histone proteins are important. Along with that, some non-histone proteins like polycomb group of proteins, they are also important, which are involved in folding of chromatin. And histone gets modified, and different modifications are there. Along with all these things in genomics, very less attention has been paid on repeated sequences, sign and line sequences, but they are involved in translocation. So in epigenome, a lot of studies have been done to show importance of these sequences. So epigenetic changes are those alterations which do not involve any kind of genetic structural changes. It happens by promoter methylation. In promoter, CpG islands are there, they get methylated, and different enzymes have been identified, DNA methylation, and some are to initiate methylation, some are for maintenance. In histone, different modifications have been identified, and non-coding RNA are also important for this. And all these things work together. So our idea is that whenever these kinds of studies are done, then all should be studied. In promoter region, whenever CG sequences in about 0.4 kb, they are 60% or 70%. These are called CpG RLN. They are unmethylated, but in tumor suppressor genes, they get methylated. So it is called hypermethylation, and this causes inactivation of tumor suppressor gene. At the same time, in some promoters, already methylation of promoter is there. By hypomethylation, they get hypomethylated, and that has been observed in oncogenes. So both ways it can work. It depends which gene you are studying. Peter Jones and Andrew Feinberg, they have been the pioneer worker in epigenetics. And they indicated that in active form, histone is acetylated, but in inactive form, deacetylation of histone starts. And when gene starts silencing, then methylation of lysine at 9 position of H3 histone, that happens for, and for stable silencing, it recruits DNA methyltransferases also. So that's how it starts. And if you see, at the time when transcription is going on, these chromosomes are quite apart, and this is shown methylation. And then gradually hypermethylation starts, shown in red, and this chromatin gets complex and complex. So those factors which are needed for gene expression, they cannot get in if it is more complex. So these steps also have been identified, and different proteins involved in that also have been identified. So in most of the cancers, globally, all DNA gets hypomethylated. But in gene-specific region, especially promoter region, that gets hypermethylation in cancer development. And histone, that shows different kinds of modifications. Now, if DNA methylation is there, then 3 situations can happen: abnormal increase, decrease, or no change. If increase is there, like it is in tumor suppressor genes, so methylation of both alleles or one allele only, or one allele and another is imprinted. So that way it can happen. When abnormal decrease is there, then like proto-oncogene activation or latent viral activation, especially in retroviral gene therapy when it was started in early 90s, then those retrovirus vectors got inactivated because of epigenetic phenomena. People thought, they did not know what is the reason. So in that also it is there, or increase the DNA rearrangements and possibly aneuploidy. And if no changes, then such changes like deamination, poor repair of methylcytosine, or chemically induced mutations, those can happen. Now, in different pathways also, epigenetics steps have been identified, and here I have shown integrin signaling network. So previously people used to study 1 or 2 or 4 or 5 genes. Now in pathways also it is possible. And similarly, a lot of research is going on in microRNA, and in 2 ways microRNAs are studied. Either microRNA profiling, that can tell whether somebody is at high risk of developing cancer or not, or that coding region of microRNA that has some changes, or nucleotide sequence changes, that also will tell. And that gene itself can get methylated or non-methylated, and that decides whether microRNA will be active or not. So post-transcription also, that regulation has been studied. In histone, these 4 histones I mentioned, and these are different modifications which have been identified in cancer. And about 6 years ago, at NIH, we initiated one program. It is called Human Epigenome Roadmap Program. The idea was the way genome was sequenced, the same way in epigenome methylation profiling or histone profiling or microRNA profiling or chromatin compactation like DNase 1 mapping, that should be done. So during that time, question came that for major modifications, high quality monoclonal antibodies are needed. So through that program we have made and we distribute to the investigators. And in histones also technologies are such that at different locations when something is changed or histone modifications are there, we can measure. And this is basically the procedure. How do you do histone modification? Like cross-linking of protein DNA complex, then lyse the cells, and then immunoprecipitation. So those things are done, and DNase hypersensitivity. One assay is also done to see that which chromatin part is accessible for transcription factors or other regulatory proteins. So in past few years, because of the interest in epigenetics, I have edited few books. Like this one was on epigenetics and its implication in cancer prevention for New York Academy of Sciences. And last year on cancer epigenetics, where more emphasis was on epidemiology. And this I did 3 years ago on cancer epidemiology, but discussing about host susceptibility factors or modifiable factors and how they are correlated genetically or epigenetically. And this year, this book will come on cancer biomarkers, in which early biomarkers we have emphasized, which we can isolate non-invasively and follow genetic, epigenetic, or biochemical changes. And 2 times I was interviewed by Nature. So I emphasized in one interview that if we compare genetics and epigenetics, so epigenetics has slight edge over genetics because epigenetic changes, they are reversible. So you can use chemicals or drugs to change. If you have mutation, deletion, or anything, you can just use for screening or follow-up. But epigenetic changes, you have that advantage. And many companies, this was for the second, they are investing like Johnson & Johnson or GlaxoSmithKline, Beecham, and AstraZeneca. All those have programs about histone inhibitors or methylation inhibitors. All those programs are in quite numbers. So that is the difference. And now, when methylation starts, then in one strand it will happen first and then on second strand. And at different steps, we can measure that. And for methylation measurement, bisulfite treatment is done, which can distinguish methylated DNA versus non-methylated DNA. And after this treatment, it is simple PCR. And this is shown that how this sulfation treatment that can change the methylated versus non-methylated region. And depending on your project, you can use either just a simple PCR method for different methylation region, or you can use methylation-specific PCR and select accordingly these primers. Or if you want to do real-time PCR for certain genes, then methyl-like technology is there. So different technologies for different objectives have come. At the same time, when you do methylation analysis, to do it quantitatively, you have to know how much is the total methylation content of the cells, or how much was the level at a specific stage, or group of gene, what was there, and profile of methylation either at a specific genes or how many number of genes are affected, or pattern of methylation in whole epigenome. And this we plan before the experiment so that we have less false negative and false positive results. In those proteins which are epigenetically regulated and are involved in gene regulation, Steve Bellin from Johns Hopkins, he is the pioneer. And he has identified which protein comes first and then comes the next and characterized those. And he has done some clinical trials also. And depending on the location, whether these are polychrome repressors or regular histone, non-histones, which stage they affect or which modification will affect, those things have also been characterized. So a lot of basic science also has been done in epigenetics. And during cancer development, at different stages, some cells are shed from tumor, and they are good sources for assay for methylation steps. So whenever you follow up the patients, you can collect samples at different times. This is a list where in different cancers, again and again, many kinds of genes have been reported which are altered epigenetically, and many reviews I have written on that. And in different categories, if you want to see whether for histone deacetylase or methyltransferase they affect, so these are the common genes which have been identified in cancer, they are affected. Now, as I mentioned that these epigenetic change, those you can reverse by chemicals, so some of these compounds are shown here. Zebulorin is very popular in phase 1, 2, 3 trial, and Jean Pierre-Issa, he is the pioneer in this field. So these are for DNA methylation or histone deacetylase. In both they have been done. They have been discovered. Saha is another compound. Some kind of side effects, as it happens with any drugs, those are there, but still they are effective. And to categorize in different groups, histone acetyl transferase or DNA methyltransferase, wherever they inhibit. So these are those agents which are either in trial, 4 have been approved by FDA, or they are being modified or used or tested. So in different categories, they have been identified very, very well. And they belong to different categories like short chain fatty acid or aliphatic compounds or cyclic tetrapeptides or benzamide, and different level, like you want to use a small amount for that, that has been done. And when this kind of research was started, histone deacetylase was only of one kind. Now we have 1, 2, 3, 8, and 11. Different kinds are there. So different companies are trying to find out how they can make it specific. So these are such inhibitors which are a novel kind of anti-cancer drugs. And many times they work in combination also, especially with PARP inhibitors, and they affect cell cycle regulation, differentiation, migration, apoptosis, etc. So now I will give you a few examples. This is a clinical trial at NCI where epigenetic modulation by DNA demethylating substance, that agent, pyroazacitidine, or valproic acid, that was given at different times and dose at day 1, 10, and 28. Then methylation and histone was measured in 55 people, and significant improvement was observed. And this was such a group of people where other treatments were not working. In another one, combination of azacitidine with valproic acid and all trans retinoic acid, that was selected. So in combination here also in different patients when it was tested, then patients started giving response. If you see, or if you want to check how many trials are going on, so at NIH, if you put clinical trials, especially at NIH, whatever are going on, that will show, and this is histone inhibitors where they are being used, whether these are solid tumors or lymphoma, leukemia, breast cancer, in different cancers. Those are being tried. Some are completed, in some still people are being enrolled, and they are being followed. And our program, we support about 31 cohorts. And cohorts are such where disease has not developed, but we enroll and collect samples every yearly or 2 years or 3 years. For example, nurses' health studies since 73, those samples have been identified, or physicians' study or multi-ethnic cohort, all that. So if we identify some marker now, since we have samples before, so we can go back and see when first marker appeared. So these are for histone inhibitors and these are for methylation inhibitors. So similarly, as you can see in different kinds, and in 51 studies those are being used. Schering-Plough and Bristol-Myers and all that, they have interest in that. So whenever in treatment you think, then you can see that epigenetic interaction is at environmental level. So that time those kind of treatment you do or inhibitors you use. Whenever genetic changes are there, then to study that those chromosomal changes or other DNA changes you see. But in terms of treatment, epigenetics is the one in which we have hope. And these are those 4 drugs which have been approved by FDA. 2 are methylation inhibitor, 2 are acetylation inhibitor. So this is not only in theories, but it is in practical. And for example, this was for a small lung cancer where epigenetic therapy was successful. Similarly, this one was for hematological tumor cells. Here also these inhibitors were very, very successful. Now in few examples I will give, like colon cancer. So these are different stages, and those genes are shown which are regulated by methylation. By genetics also in colon cancer, a lot of things have been done. So our focus is to exploit that. And in this also, in large number of sample, one phenomenon was identified in colon cancer especially. It is called CIMP, or CpG island methylator phenotype. A group of patients, in colon cancer, if you know, left side of colon behave differently than right side. So many times people did not know which is for high risk or low risk. And genetic or biochemical or proteomic or any test could not distinguish, but then CIMP phenotype could distinguish those. And in that, integration of genetics and epigenetics both was studied. And as you see here, KRAS mutation is very common in colon cancer, BRAF mutation also. Along with that, microsatellite instability, high or low, or these colors are shown for methylation, hypermethylation. So its combination was used, and based on that, they could identify a group of genes or the way it can be identified. If someone is at MSI high, or it has BRFF mutation and hypermethylation and CIMP phenotype, then they are at very high risk of colon cancer. In AML, ALL, just based on methylation profiling, those populations could be identified or distinguished. In lung cancer, some genes are regulated by methylation. Along with that, if you include annexin-1 or annexin-2 or some genomic markers, then you get more sensitivity. And Steve Belinsky, he has used sputum for screening or follow-up of treatment, and he uses these markers. Now, asbestos I mentioned about mesothelioma. So for a number of genes, like 803 cancer-related genes simultaneously could be followed for methylation profiling. Now comes esophageal cancer. So Barrett esophagus, that is the early stage of esophagus. So 20 genes simultaneously. In the same patient, samples were collected at different stages. So as you see, level of methylation in different genes, it is different. So this can be quantitated also. Percentage methylated region we can calculate. And sometimes that methylation profiling you can correlate with survival also, based on hyper or hypo. In pancreatic cancer, this cancer and ovarian cancer, these are such where incidence and prevalence or mortality rate, that is almost the same. By the time you detect in 1 or 2 years, it is too late. So early markers are not there. But if in pancreatic cancer, if we know somebody is alcoholic, long-standing diabetes is there, KRAS mutation is there, then P16 and P14, if they are methylated, then those people are likely to develop pancreatic cancer. So that also implies that whenever patients are enrolled, then we take patient history and their exposure history or genetic background, and all that is very useful Here in this case, lifestyle like alcoholism or diabetes or other diseases what are there. So those are sometimes very useful. In breast cancer also, lot of genes were followed simultaneously, and in that treatment was also followed along with methylation profiling. And in breast cancer, their node levels or methylation profiling for different genes that could distinguish high-risk population versus low-risk population. You heard about cervical cancer before some time. In about 15 to 20% cancer, infectious agents are involved, whether it is HPV, HBV, or HHV8 in Kaposi sarcoma, hepatitis B, C, all this in liver cancer. So those are there, and especially latency-associated nuclear antigen or Epstein-Barr virus nuclear antigen. This was in nasopharyngeal carcinoma. This was in general. Those were identified which were regulated epigenetically. And those were the genes basically they help in getting integrated that virus. Many times whenever viral load is measured, that free virus you measure or sometime integrated, but both should be done. And in many cases, it has been observed that these viruses are affecting different sites. HPV initially, it was only cervix. Now in lung, prostate, colon, many places this is there. And in last few years, complete methylome of HPV, EBV, HBV, that has been done by Manuel Estler. And why we do these kind of studies? Because when there are no symptoms, then also we can tell who is at high risk or not. And those kind of approaches, like previous speaker told about vaccine, so those kinds of approaches can be done. And here it is shown that like early genes of cervical cancer, this HPV, or late genes, how much methylation is there, when it is started, all those things we will know, and we know about at which stage this is there and how much methylation is there. So those have been very well characterized. Now in hepatocellular carcinoma, sometimes these kinds of observations also we see, like this one, this gene, that is regulated epigenetically only in one population, not in others. So these kinds of observations also come. Otherwise, common genes in hepatocellular carcinoma, that liver cancer, that exist. In oral cancer and head and neck cancer also, some genes have been identified which again and again different groups they report. In immunology, lot of epigenetic regulation in B cells and T cell development and their role, that is being studied. Now in prostate cancer, whenever level is less than 4 nanogram, doctor cannot tell whether person should be treated or not, that will develop a cancer or not. In that aspect, GST-P1 methylation is coming up as a very strong marker. So PSA levels and GST-P1 in the same patients were also followed. And after some time, one company introduced a kit, and Epigenomics and Quest Diagnostic, they acquired that. So this kit is out in which prostate cancer is detected by GST-P1 methylation also. In bladder cancer, we emphasize anytime you detect, diagnose, or for prognosis or follow-up or survival, the sample should be non-invasively collected. So in urine, whatever exfoliated cells come, in this case, laminated gene, that laminin, that was followed for methylation, and bladder cancer could be followed. Now, many times multiple cancers are also in patients, or one cancer is developed in one site and then it metastasizes in other organ site also. So these methylation profiling in different cancers in the same person can also be followed, and Peter Laird in University of Southern California, he is the pioneer in this. Now diet plays a major role, and it has been observed that many of these DNA demethylating properties or histone modification properties, they are present in diet. And some animal models also have been identified where just based on diet, a mouse or rat that will develop diabetes or cancer and all that. So a lot of things have been done. And in cancer field especially, people think that 30 to 40% of cancers you can prevent if you change diet and lifestyle. So those kinds of studies are also going on. Now these are few examples and active compounds I have shown, which are called nutraceuticals, which have either histone deacetylating property or demethylating properties. And if you see methylation inhibitors in tomato, apple, tomato lycopene, or these are present, and some have methylation and histone deacetylation inhibitor both. So those have been identified, and if you see those regular vegetables or other compounds, compounds which have been identified in food and food, their active components, they have been characterized for their epigenetic inhibiting properties. And at different steps they work, and this regulation I already mentioned that in open form, this gene expression work where that has been identified. And these are different compounds from these food, and sometimes histone at a specific position that will be modified more and sometimes less. So characterization has been done to quite an extent. And if you hear about that alternative medicine, in that these compounds are being tried. And those food have also been identified which are common for both or alone. Now in liver cancer, just by changing the diet, such kind of models have been proposed, acquired, where you can develop same kind of features which are in animal models, and no carcinogen or other agent was used in this case. Now regarding what technology we should use for epigenome, so sometimes we need resolution at low level, sometimes we need at high level. Technologies exist for both levels, so that decision you have to make that which way you want to do. And in microRNA, as you know, this field is growing up very much. And in cancer specific, those have been identified, and these all steps have been characterized. And in cancer, for different cancers now, different microRNAs, these are the number of microRNAs identified in different cancers. So usually their profiling works. And the way we recommend is that whenever you have a sample, so don't do one test, but do many tests. MicroRNA profiling, methylation profiling, or histone changes, or if you have genetic information also. So that has been very useful. Now, little bit about NIH roadmap. So these are such programs in which epigenome was one of them. Microbiome was also that. Many times, some mechanisms are such they are for many diseases. And epigenetics was such. So NIH thought that many institutes are involved in that. So they decided to follow up whole epigenome, all those 4 modifications. And these are different diseases where epigenetic regulation was very well established. And what happens that in this, all institute will contribute money for that, and about 1.5% of total budget comes to these roadmap programs. One metabolome is also going on, or others are also there. And what it supplies, it gives us some time and such kind of resources so that you can try some high risk but emerging scientific opportunity, and then different institutes can take up whatever results come. And this is one example where in 2008 we started, and up to 2015 our commitment is there for $219 million, and now we are extending it for 3 more years, because now we are at this stage that we have different populations, some compounds have come which are promising. So those kinds of studies can be done, as well as lot of basic sciences also being done in there. After we started this roadmap program, then in Europe they started IHEC program. It's called International Human Epigenome Consortium. So their idea was that without overlapping, whatever is left, that they want to do. So what they did, from disease samples, they started characterizing that. The way in NCI we have TCGA program, Cancer Genomic Anatomy program, in which well-characterized tumors are taken, and then they are characterized how much genetic mutations are there, how much methylation profiling changes and all that, high quality. Similarly, Europe is trying for different diseases, and here partners are industrial partners also. And along with human, they are working on animal models also. That is the main difference. But we are part of that also as representative to check there is no overlap and information is complementary. So we had initial meeting of IHEC. These are the countries in which they are involved in IHEC program, and different funding agencies, they are contributing as well as companies. Wellcome Trust is the main, or European Scientific Foundation or Commission, and Epigenetics, Genoscope, Novartis, all those are there. And when it was started, the Nature and Science both covered this IHEC program. And AACR, our American Association of Cancer Research, they are very much interested. They always are in our meetings. So ideally, when a patient comes, we think that when they do other analysis, they should do methylation, histone, and microRNA profiling also. It may take few more years. And as you know, again and again about personalized medicine, everybody responds differently to treatment. And now we are in a position that at least we can follow up those. So genetics, epigenetics, all these we can follow, so that will be very good. So in tumorigenesis also, we have to think about these inhibitors as well as combined therapy, then only we can achieve something. And these are the investigators who are thinking about using how to use these epigenetic approaches to identify ethnic group special or specific factors, or identifying how we can use in combination these markers with others. Because some cancers are very much race correlated, or they are at high risk of developing cancer, and in US different people have come from different countries, so these are the ones who are trying to address these questions. And one term also time to time you will hear, this is called EVAS, epigenome-wide association studies. So the way GWAS or genome-wide association studies were done, similarly 3-4 EVAS also have been done. And similarly, I participate in human metabolomics also. So there we are trying to find out that in healthy person, what are the metabolite pattern, and disease-associated metabolites are there. So that in few years we'll have information, already genetic information we have, epigenomic information and metabolomics. That information, then we can put together. And in metabolomics, since metabolites, after they come out, they are very much stable. In urine, you can measure, and they reflect which pathway was associated, so that also will be very, very useful. So anytime any project is done, you think about phenotype or genotype, but in between, this epigenotype is also there, which you should consider. And these are the collaborators, which time to time we take their help or their advice. And I will take any question. Yes. Yeah, and that is the place where our programs support. Like, okay, the way research works in US, most of the time from different universities, investigators will propose, okay, we want to do that. So the way you are reading, somebody will propose, okay, I want to study oncogenes, somebody tumor suppressor genes. That they do and they report, which is good to get information. In our program, we want to know actually, since all this information is known, what is happening for all these. So we write our contracts. So we assign certain amount of time and money and identify people who can do that. And they do the research. So we give them samples at different times and ask them that study how much is going on. So in different stages, different steps, which one is more, which one is high, so that information is there. But those studies are not in many numbers, but those also exist. So if you put for histone markers and methylation markers like that together, those kinds of research if you do, then those papers will come. But that is being studied. And Yeah, that can change, but it is not necessary that all genes are affected all the time. First of all, as I was emphasizing, environment. So environment, and as these 12 pathways are being characterized, so all are not affected, some are affected. And they are also environmental agent specific. So in that pathway, what genes are there, those are affected. And similarly, at initial stages, that is there. Once a secondary cancer comes, or sometime diabetes and cancer is there, those are there, then only you see that other genes are also affected.
That'll do it. Thank you very
Okay,
much.
thank you.